Hey guys, it's your favorite YouTube content creator slash Biology 9 teacher with another section notes. This one is on protein synthesis. Now we're moving into the second stage of protein synthesis, and it's called translation. So let's get going. Here are our ICANN statements so far. The big check marks we've completed. They're all the way done. So those two little check marks we're still kind of working on, and then we're also going to be working on ICANN statement number 6. So in this section notes, we'll describe how complementary base pairing guides translation. We've already done it for replication and transcription. We'll outline the flow of genetic information in cells from DNA to a protein. So we've started that process. We've gone from DNA to the mRNA. Now we need to make the next steps to tRNA and the protein, something we worked on in the protein synthesis activity. And then describe the roles of mRNA, rRNA, and tRNA. We'll be talking about how those three types of RNA are involved in the translation process. Let's go ahead and do some review. And for this review, in class, we would normally talk about it with our table group. But what I'd like to do instead is I'll put up these questions, and I want you just to pause the video and see if you can answer them. So our very first question is the differences between the nucleotides for DNA and RNA. The second one is about the enzymes that are involved in replication. What does helicase, polymerase, the DNA polymerase, and then RNA polymerase do in transcription? What is replication? Where does it occur? What is transcription? Where does it occur? So just kind of knowing what they are and where they happen. And then lastly, what is a gene? Pause the video and see if you can answer these questions, and then we will go over them together. So hopefully you paused. Let's go over these. If you know these 4 questions, you're in a really good place for the content so far in this unit. So we know that the 3 parts of a DNA nucleotide are a deoxyribose carbohydrate sugar. We know that there's a phosphate group, which kind of attaches the strands together, the nucleotides to make up the strands. And then we have a nitrogen base, which we have 4 of them: adenine, thymine, cytosine, and guanine. RNA nucleotides are pretty much the same, but also different. So there is no deoxyribose, in its place is ribose. And then the phosphate group is there, but the nitrogen bases, instead of thymine, there is uracil. For number 2, we know that enzymes do help those processes happen. Helicase is the enzyme that's going to break apart the hydrogen bonds in the DNA molecule, opening up a replication fork. And the DNA polymerase is the main enzyme that will begin to attach free nucleotides by reading the nucleotides of the parent strand and base pairing them together. RNA polymerase, that's the enzyme that's doing pretty much the same thing, but instead of producing DNA, it's producing RNA during transcription. Replication is the copying of DNA, and it occurs in the nucleus. I'm on to number 3. Transcription is making a copy of DNA. A copy of the DNA template in the form of mRNA, and it also occurs in the nucleus. And then number 4, a gene is just a section of DNA that codes for a protein. Where are genes found? Well, they are found in the DNA. So if you're good with those questions, you're in a really good spot. If not, I would definitely go back and review those. We talked about how producing a protein, and we saw it in our activity, but how producing a protein is kind of done in a code. And the code is not harder than this. Here's a rebus puzzle. And here is another rebus puzzle. Don't know if you like the rebus puzzles. I do. So that first one on the left is an inside job. See, N is inside job. And the second one is win with ease. My favorite one, win with ease. I promise you, translation is not that difficult. We've already started the process of making the protein with transcription. So we know that in order to make our protein, we made the analogy that's kind of like making a cake. And so the first step is we have to copy the recipe or copy the instructions. All right, step 2. We're going to use that mRNA copy to begin making our protein. In our cake analogy, this would be like getting together all of our ingredients. So we're going to gather up, there's our final cake, but we're going to gather up our recipe. We're going to get our eggs, our flour, our chocolate, our vanilla, our oil, our sugar, our milk, whatever we need. And we're going to set them all out so that we can start to produce our protein or our cake. And again, this is kind of what we're going for. We started with our recipe and our instructions. Now we have our ingredients, and we're going to use that recipe card. In biology, it's the mRNA strand to make our cake or our protein. Okay, translation is the process of decoding the mRNA to assemble the protein. Now I'm just going to put this note up. It's pretty big. I'll let you guys write it. I'll stop talking for a minute. Get myself a drink. Have you finished yet? It's a big bullet, I know. Did you write it all? I mean, seriously, if you wrote it all, stop. Just stop. Because it literally says occurs in the cell structure that is the site of protein synthesis, but you should know what that is. So instead of writing all of this, you should just write the word, which means it does occur in ribosomes. So translation is the first thing that we're learning in this unit that does not occur in the nucleus. Replication, nucleus. Transcription, nucleus. But after transcription, that mRNA molecule is going to leave through a nuclear pore. It's going to leave the nucleus, and it's going to head out to a ribosome. It can go to a ribosome that's attached to the rough ER, or it can go to just a free ribosome that's kind of hanging out in the cytoplasm. Now, in order to decode the mRNA, a codon is read. And a codon are just 3 consecutive nucleotides that specify for an amino acid. We looked at these in our activity. We looked at the codon AUG. All of our proteins that we made in the activity started with AUG. That's a codon. Amino acids are the monomers of a protein. We should know that though. I only put that definition in that bullet because amino acid was used in the definition for codon. But we should know what that is. I'm in my classroom today doing this video instead of being at home. And I'm pointing up right now to the proteins poster, which says the monomer of proteins is an amino acid. So that comes all the way back that goes all the way back to Unit 1. We should know what the monomer of a protein is, but just in case you forgot, or we do have some new people that now need to know that. If you need more time with the slide, go ahead and pause. It's a really important slide. Like you 100% need to know what a codon is. You 100% need to know that amino acids are the monomers of a protein and are made one by one from reading a codon. Okay, I have gone ahead and turned off the camera for this slide because I need to be able to use the entire slide for the next couple of slides. I'd like to introduce everybody to this wonderful, wonderful diagram. This thing right here. This is the genetic code wheel. There are different forms of this. I prefer the wheel. I know that I talked to Dr. Mitchell. She prefers the chart. But I like the wheel. I think it's easy to use, and I'm going to teach you guys how to use it today. So let's suppose that we have the mRNA message. We've already transcribed our DNA into mRNA. Let's suppose we have the mRNA message AUG. And so to use the wheel, what we do is we start from the inside and work out. We're going to begin with the letter A, and then we just kind of go outwards. My next letter is U, and then I have a G. Whatever the word is here that lines up with that AUG in this case, or those 3 letters, that codon, that is our amino acid that we're putting in place. And so our first amino acid that we're using is methionine. There it is. That's our first amino acid. That is also the start codon. So not only does AUG signal for methionine, it also signals to start making the protein, which means all proteins, every protein that we have, all 20,000 or so in our bodies, the first amino acid that was put into place was methionine, always, because it's also the start codon. Methionine could be somewhere in the middle of the protein as well, because an AUG could come up later, but it wouldn't start twice. If it's already started once, then methionine could also just be in the middle. Let's suppose that our next codon is CAG, and so we start with C here in the middle, and then we go to A, and then we go to G. So our next amino acid is glutamine, and we can abbreviate glutamine. We can abbreviate all of these with just the first 3 letters. We just have to be careful because there's a glutamine here, then up here there's also glutamic acid. So if I had glutamic acid, I would write probably glue acid just to differentiate between the two. Okay, our next one is GUU. So I start with G, that's the first letter in my codon, and then I go to U, and then I go to U, and so I'm going to put in place here a valine, VAL. And then last is UGA. So I have U, and then I have G, and then I have A, and that's a stop. So stop is not the name of amino acid. All these are like phenylalanine and leucine, serine. That would be really weird if it was just stop. Stop is the stop codon. UGA is one of the 3 stop codons that signal the cell to stop making a protein. So anytime it's UGA, UAA, or UAG, there's a stop. It's a little bit different than what we did in our activity. In our protein synthesis activity, there was only 1 stop codon. That was just to make it easier in designing the activity. But yeah, there are 3 different ways, 3 different codons for stop. Hope that makes a little bit of sense. We're going to try another one. So for this one, we've got a little bit more to do. And so what we're going to do is we're going to transcribe this DNA message together, and then I want you to translate it to see if you can do it using the codon wheel. So let's go ahead and do transcription first. We're going to take TAC. We know that TAC is going to become A, and I'm sorry, my handwriting, I'm trying to write with the mouse. It's not that easy. A, U, G. GAG will be C, U, C. I'm going to jump down here to the bottom. CTG, just to have space, is G, A, oh, that was an awful A, GAC. And then ACT would be UGA. All right. I'm going to go ahead and push pause. I want you to pause the video as well. And I want you to try to use the codon wheel. So use this to see if you can translate the mRNA codons to the correct amino acids. Pause the video, try it now in your notes, and then when you push play, I'll have the answers up so you can see how you did. Okay, hopefully you've gone through it. You can see kind of my annotations on the codon wheel. For methionine, I used, get the laser pointer, I used AUG to get methionine. For CUC, those are my underlines here in blue. C to U to C, that's leucine. I put leucine right here. GAC, GAC is aspartic acid. I had to write out acid to differentiate between aspartic acid and asparagine. And then I had UGA, which is our stop again. There are only 20 amino acids, which means that some codons will code for the same amino acid. You may have noticed this, that GCU is alanine, but so is GCG, and so is GCA, and so is GCC. So there are different ways to get the same amino acid. There are 64 possible codons and only 20 amino acids. So we could have a mutation and then nothing happened because the same amino acid gets put in place. We'll talk about mutations a little bit more Let's continue our discussion on translation. All right, back to finishing up our notes after doing the translation practice. One more thing about the amino acids. There are 20 of them. Our bodies can make 11, and we have to get 9 from food that we eat. They're called essential amino acids. So it's important to have a balanced diet to make sure you have all of the building blocks that are necessary to make proteins. And so let's go ahead and finish that process now. We've got a few more notes that we're going to take. Okay, the codon wheel is great because it does some steps for us, but as we know from our protein synthesis activity, real cells don't have a codon wheel. And so these are the 2 steps we've done so far. We've done transcription, we've done the beginning of translation. We're still using our cake analogy. So our final step is to take amino acids and attach them together using tRNA anticodons and matching them with the mRNA codons. And again, what it says underneath the camera screen there is that the codon wheel does this for us, which is great. In our activity, you had to go and find the tRNA molecules that were floating around the classroom. You had to go figure out, well, I have this codon, I have to match it with this anticodon. How can I do that? Oh, I've got to match it with this anticodon and see what word is attached to that tRNA molecule. The codon wheel does that for us. Awesome, but in real cells, it doesn't happen that way. So it's like taking our cake and putting on, you know, I don't really love chocolate, but I love peanut butter, or I love a nice peanut butter cake, maybe some chocolate chips to make our chocolate cake even better. This is where we're taking our final ingredients and we're attaching them all together. We're putting them all in place. And then that cake can go, we can package it up in a vesicle. We could ship it off to where it needs to go using the Golgi apparatus. Maybe we'll put it in a box and send it to a relative, or share it with some friends or family. Then that protein or that cake can go to wherever it's supposed to go. So this is our final step. Now I use some words in step 3 that I think it's important that we define. So we know that the protein is controlled by the start and stop codons. The only reason I put this in a red box is because AUG is the only codon you have to memorize. You will have the codon wheel on every assessment, including the Unit 5 Quiz-Am, including the final, if there is translation questions on the final. So you just have to know how to use the wheel. You don't have to memorize it, but you have to memorize AUG, because the wheel does not say that AUG is the start codon. It does say it's methionine. There are 3 ways to stop. And as we said, the tRNA is matching anticodon to codon. So the codon is on the mRNA, and the anticodon is on the tRNA. Where is our RNA in all this? Well, our RNA is a part of the ribosome. It stands for ribosomal RNA. The ribosome is made up of our RNA and some protein that never really gets used for anything other than being a part of the ribosome. Attached to every tRNA molecule, as we saw in the activity, is an amino acid. So when you were going around in the protein synthesis activity, we were using words as amino acids. You would find the anticodon, the tRNA, and the word all around the room. If you need more time for this slide, go ahead and push pause. Otherwise, we're going to keep going. You can see the whole translation process in this diagram here, and there's some really important things, and you had to label a similar diagram on your activity. So we have our mRNA molecule right here, and on our mRNA molecule we have codons, so you can see like GAU is a codon and your CA. This big green thing on the outside, this kind of bluish turquoise thing, is our ribosome. And then we have our tRNA molecules. Some tRNA molecules don't have an amino acid like this first one because the amino acid's already gone on to the polypeptide, which is another name for a protein. But it does have our anticodons, and we can figure out what these are. If we looked at our codon wheel and looked at ACG, then we could figure out what this amino acid is, and maybe you could do that for practice. CGU, we could figure out what this amino acid is. Now what I'd like for you to do is pause this video and exit out, and I want you to find this video. This video the link will be right below it on Moodle, and play this video because it's the translation process happening in real time. You're going to see one hemoglobin protein made in the video, and it shows you about how fast this whole process is happening in your cells, which is just incredible. So pause this video, exit out, and then once you've watched that video, come back for the summary, and I'll see you there. I'll have the camera on for that last slide. Okay, if there are no questions for the good of the cause, our summary today is an I wonder question, because we've learned a lot. We've learned about how DNA fits into the cell. We've learned about how it's replicated, how it gets converted into mRNA to make a protein. So what are you wondering for your summary question? My question, and it gets covered up, but I'll explain it. I'm wondering which eukaryotic organisms, I don't want to involve prokaryotes because logically they would not have as much DNA as a eukaryote. So which eukaryotic organisms have the most base pairs? I told you guys that humans have 6 billion total nucleotides. That's 3 billion base pairs. Is that the most? Do we have the most DNA for eukaryotes? And then which organisms have the least? Are there organisms that might surprise us that we would think are pretty complex that have a lot of bases? So those are my questions. You hopefully will write your own, and I will see you guys. We'll answer this in the next class. We'll answer this I wonder question, and I will see you guys then. Take care.